So once again, my name is Ieva, and I'm a third-year PhD student at the Lithuanian University of Health Sciences in Department of Genetics and Molecular Medicine. I have studied medical and veterinary genetics for my bachelor's and later on my master's degree, and now I'm a PhD student. And my PhD research is focused on cancer genetics and epigenetics. Moreover, I am also involved in research of male infertility. And to reach my goals and improve knowledge and skills, I was on a couple of scientific traineeships at Copenhagen University Hospital in Denmark for in total 9 months. And recently, I was actually awarded with Baltic American Freedom Foundation scholarship for a one-year traineeship at Center for Embryonic Cell and Gene Therapy at the Oregon Health and Science University in the United States. And I will be going there this summer. So now let's hear about Dovydas.
So hello again, everybody. As Julia said, my name is Dovydas. I am a second-year PhD student at Lithuanian University of Health Sciences, Institute of Pharmaceutical Technologies, and also a junior researcher at Institute of Cardiology. My main topic of research revolves around various cellular processes, including intercellular communication by extracellular vesicles, changes of gene expression during various conditions, and biomarker identification. I work with several scientific technologies like fluorescent microscopy, protein analysis, recombinant biotechnology, gene expression assays, and as well as next generation sequencing. And same as Ieva, I have finished this genetic program 5 years ago, done my master's at Lithuanian University of Health Sciences as well, and I still continue my work here with several research groups. So the first topic, why did we choose Lithuanian University of Health Sciences? When you are at school, choosing the right higher education is a difficult task. I know that because I've been there myself. For me personally, this decision to study in genetic program was rather spontaneous because I was choosing between medical practice and a scientific field. 9 years ago, when I was still at school, genetics seemed as a new, innovative and progressive field with a huge growth potential. So I took my chances and went into a rather unknown field and applied to Lithuanian University of Health Sciences. Why here and why not Vilnius University? Both of the universities are considered prestigious institutions in Lithuania. However, the location of Lithuanian University of Health Sciences and a good feedback from my older friends helped me to make my mind. Lithuanian University of Health Sciences is located in Kaunas, which is in the middle of Lithuania, so it makes traveling around very easy. You can reach your colleagues in Vilnius, you can reach your colleagues in Klaipėda. It saves time. It is a very good geographical location. So, Ieva, do you have a word what it is like to be a student here?
I can actually only agree of what you just said. And I think that it's very important. Well, if you want to be a successful student, I mean, it's important that you should not only be eager to learn the theoretical stuff, but also engage in laboratory practice, like a lot of it, because you can apply your knowledge to practice and see how it works. And you see it for yourself in various experiments. And so it is also a very good idea to look for a traineeship at the lab, which is focused on the topics that you are really interested in. So, and the final work of every student of medical and veterinary genetics is to prepare a mini version of scientific work where you perform experiments, describe them, and the obtained results as well in a very short paper, and after that you have to defend it in public. And so I think that if you start learning early on, you will have a solid foundation for your future career, and it doesn't matter if it's in diagnostic or in research lab.
So to add a little bit more to what Ieva said, in my opinion, being a student at Lithuanian University of Health Sciences is a privilege and a responsibility at the same time. University has a very nice infrastructure for students. For example, we have a very modern library which is open 24/7 with essential up-to-date literature and subscription to the latest scientific databases. There are also a gym for students, cafeteria, science clubs, choir, and dance groups. So I think there are enough activities outside of learning. You can find some friends, you can socialize, you can join your colleagues in different activities. Of course, if you are into sports, university has sports teams like basketball, volleyball. I'm not into sports, so I cannot provide not a lot of information here. For me, what is very nice, personally, university has a mentoring system, especially for international students, where the mentor is assigned to a group of students and they help you to get around, to know the city, to know university facilities, which is really helpful if you are a freshman. So again, to add a little bit more to what Ieva said, apart all extra curriculum activities, MVG studies require time, and you have to put yourself into it. And the good thing is that if you want to be really good at your field, you can do it. It is up to you. University provides you with all the material that is necessary to reach your goals. So this program consists of genetics, biotechnology, biology. In addition to that, this program introduces you to medical fields. Like in the first year, you usually start with human and animal anatomy, and the studies take place in real projectoriums where real tissues, real organs, real parts of animal and human bodies are examined, just to get you that deep knowledge of how we are made as living creatures. So going deeper, one of the most important studies of the program, in my opinion, is histology and embryology, because during our workday and later research, we spent a lot of time doing microscopy slides. And during this subject, you cover a lot of microscopy. And I liked it very much. So in later years, you will go even deeper and cover basic cellular processes in molecular level, which was for me, again, very interesting part. And I continue my work in the molecular part of the biology and genetics. So again, what Ieva mentioned about the practice and the final thesis, in my opinion, one of the strongest parts of Lithuanian University of Health Sciences and MVG program is that university has several very strong research institutes, such as Neuroscience Institute, Institute for Digestive Research, Institute of Cardiology, Institute of Oncology, and others. The good thing is that most of the staff is English speaking, and the institutes and laboratories are willing to accept interns who are keen for science. The program has mandatory internship period from 3 to 4 months, so you can apply what you learned during your studies in real research and joining ongoing projects, which is really nice. And this happened for me as well. I joined a real ongoing research and tried to make a little bit of difference in the field. So during the second half of second year, of the program, you will be introduced to the main topics of the final thesis. So as Ieva mentioned, you will have to prepare a small research work of your own together with your supervisor. And the good thing is that this project might end up as scientific publications or develop into further studies. So while studying here, I really like the continuity of the work. So when you start something, you can work on it further, prepare project based on your research, and publish some articles. So one of the most important questions that everybody has, what to do after graduation, what I can do with this degree. Is it any good? So in my opinion, yes, it is. It is pretty good, actually. So there are three main paths for MBG graduate, and it is biotechnology, medical testing laboratories, and research. And if you decide to study here, my suggestion, try all the fields after graduate. Decide what you like, and then move on, go into it. Become the specialist of your field. Biomedical technician is the specialty that you get here, is an all-around specialization for a health science specialist, which provide a good insight about this field and gives a good baggage of knowledge to be a competitive specialist. So to sum it up, I wrote for myself this, that university provides us, the students, with the tools that are necessary to build on our future. But the students must never forget that they are the builders, and the success of their career are in their hands. So, Ieva, do you have anything to add here?
Actually, I think you explained everything in full detail, so I will not add anything, but we have a video prepared for today, and we will show you the basics of in the genetics lab. So that will be the DNA purification, PCR, and electrophoresis. And so we will kindly ask a statist to please show the video and we will comment a little bit during the video so that you understand the basic steps. So to begin with, we have some prepared tissue material in the tube, and we will perform DNA extraction using silica columns. So first step is to pre-lyse the sample by using provided buffer and proteinase K. It takes approximately 3 hours in real time, but it's not going to take that long during the video. So, and also if you have more tissue, it can take longer, for example, overnight, because you need to incubate it so that the proteins are digested with the enzyme you add. So here is the incubation for 3 hours that I just mentioned. And then once the incubation is complete, we have to lyse the sample with adding a buffer and incubating at 70 degrees for just 10 minutes. And in this step, the membrane of the nucleus is removed to make the DNA accessible for further extraction. And then the next is to wash DNA with ethanol. It precipitates the DNA, and the DNA might become visible if you have large volumes of starting material. In our case, it didn't become visible because we had only just a little bit of tissue. And then in step 4, we have to bind DNA to the columns. So that's just basically transferring DNA from a tube to a column. And we will wash it by adding different buffers and centrifuging a couple of times. So this step helps to clean DNA without losing it. And the washing is very important step in this procedure because you usually need very pure DNA for many experiments. So you have to remove various additives that are in the DNA, for example, proteins or salts. So you have to wash the membrane and centrifuge it. And that's the second wash that I mentioned. So we add another buffer once again and centrifuge it. Centrifugation takes 1 minute in real time. It's usually the time that is used for such purpose, but we just had to cut it so you won't be bored looking at the centrifuge. And we remove the liquid from the column and centrifuge it again to dry the membrane. This is really important to remove all of the ethanol so it won't get into further analysis. And once the ethanol is removed, we will place the column into a clean tube and elute our DNA with elution buffer, meaning that the DNA from the column now will be in the tube. So here is elution buffer. It's usually preheated. And we place it on a column and keep it for approximately like from 1 to 10 minutes. And after that, one step actually of centrifugation, and you have the DNA. Okay, so now the DNA is in the tube. So we have to label it so we know what's in there. And here it is, the DNA is ready. So you can do a lot of experiments with extracted DNA. However, it is really important and you should measure the concentration first, because in a lot of further analysis, you need to know what's the exact concentration of the DNA. So you have to calibrate the machine with the elution buffer that you used for eluting your DNA. And once this is done, you have to put your sample on the machine. It's usually 1 to 2 microliters. And when it measures, this is a spectrophotometer, and it tells you the concentration and the purity of the DNA. And well, it looks pretty good, the DNA concentration, and it's good for further analysis. And Dovydas will take over from now on too.
So the polymerase chain reaction is a very important tool to use during our work. During this process, we amplify specific regions of the DNA. We make a bunch, actually a lot of copies of the same fragment, of the same sequence of nucleotides to analyze the mutations, polymorphism, and other interesting stuff inside the DNA chain. So here we are preparing the polymerase chain reaction mix, which consists of salt buffers, specific primers. Primers are used for generating the fragment, and DNA polymerase enzyme, which synthesize the fragment using nucleotides provided in the buffer. We distribute this master mix into the sample tubes. Usually we prepare more than one reaction to test different conditions of the PCR reaction. During this program, you will learn a lot about PCR because it's our everyday's bread and butter. Basically, every student has to do this. Every student has to learn because now this became as a standard procedure in molecular genetics lab, and it is used everywhere. PCR is used for coronavirus testing, for example. So we distribute the DNA sample into the 5 tubes, which contain master mix. In this case, we used 5 tubes because we will test different PCR temperatures in each tube to decide which one is the best. So we briefly centrifuge all the liquid to put it down at the bottom of the tubes. So there are no bubbles, so the reaction can be performed efficiently. And then we will put everything into the PCR device and start the reaction. So the PCR usually takes around 1.5 hours, depending on the conditions. After the PCR, we will run the agarose gel to visualize the PCR fragments that were synthesized during the reaction. Here we are preparing the gel. So it is the basic agarose electrophoresis gel. If I remember correctly, it is 0.7%. So our fragment length should be around 700. So this percentage is fine. So we mix the gel with the buffer, and then we put it into microwave to boil the agarose. And when the agarose solidifies, the gel polymerizes and it becomes a hard material with pores. And this technique is actually a very basic technique, a very simple, and we use it a lot to check for errors, to check the quality of our DNA, to check for amplification products. So the gel is run in the electrophoresis bath, which is filled with buffer. Here we mix the sample with the loading dye because DNA has to be brought at the bottom of the gel well. And the dye helps us to see where our product is. And we do this for all of the 5 samples. Probably what you saw early and what you are seeing now, the big part of our work is transferring small amounts of liquid from one place to another. So we put on the lid and then we start the program. And then because of its charge, DNA moves horizontally in the gel, and DNA separates by its size and charge. So the run usually takes around 45 minutes, and then we visualize the gel under the UV light. And there we can see the relative amount of DNA in the gel and the size of the fragment, which is really important. To measure the size, we use the DNA ladder, which is on the left, and our fragment is these 5 bands from the second well to the last one. So, thank you for watching.